So yeah, I want to make 2 points during this talk. One is that if you think about life in terms of lineages and clades, we actually know far more than we know at the species level. That's actually very good news. And the second one is that, as the previous and subsequent talks will illustrate, access to high-quality genomic tissues is rate limiting in biodiversity genomics. And the iBOL 2 community is one of the most powerful forces for collecting genomic material that exists on Earth. So there's a real possibility for us to build a bridge to this very exciting field of biodiversity genomics and contribute to it. It's not only exciting, it might actually make us wealthy. So, let's see, go like that. So I run something called the Global Genome Initiative, which wants to preserve and understand the genomic diversity of life, all of life. And our sort of characterization of the landscape is where we're at now is with regard to finding tissues, they're hard to find. You don't know who has them. They're obviously often of ambiguous quality, and they're ambiguously owned by individual PIs. You have a Revco in freezer in your office, and that's it. After, we want those genomic samples in institutional biorepositories, so they're supported institutionally rather than by individual PIs. And they also have to follow best practices and especially international treaties like Nagoya, which is a fact for all of us we have to adapt to. We are currently doing sort of boutique sequencing of a relatively few genomes. You can use phylogenetics to do a coordinated, thoughtful sequencing using the tree of life as a guide. That will advance genomic knowledge of life faster than other techniques. This is something that's very relevant to barcoding. We're still often in the, not this community, but we're still often in the situation where we have phenotypic, expert-limited taxonomy. You have to find a taxonomist, put it under their nose, and they tell you what it is. If we pull this off, we might be able to get sort of mesoscale IDs of most organisms anywhere within a few years. That means, mesoscale means at the level of genus and family. So that's really the promise in a way that barcoding wants to offer, but barcoding usually prioritizes species. If you go down a little bit, it becomes much more feasible, something you could do as a priority in the first few years, say, of Paul's project they talked about yesterday. And then obviously all of this adds to also what Paul was talking about, sort of environmental biology, biosurveillance, evolution, all of that. We want precise, scalable, cheap tools that gets the expert taxonomist out of the workflow so that the taxonomic impediment, as it's called, disappears. So this is how I think about the 4 ways to be a genomic sample. Warm living is the best. That's like the maple trees out there. That's what you want. You want critters out in the wild, organisms living their lives, in perpetuity. The next best is sort of the cold living. Those are frozen samples that are intended to be alive when they come out of freezing. So those are seed banks, cell cultures, some zoos, things like that. Less good is what I call the cold dead. That's essentially what we do, which is we put samples in freezers, but we don't expect them to come out alive. On the other hand, cryobiologists tell me that any piece of living tissue plunged into liquid nitrogen without cryoprotectants, somewhere between, say, 10 and 20% of the cells will be living when you take it out and do the first thaw. So things don't actually die at liquid nitrogen temperatures completely. And then, of course, the last thing we've been doing since Linnaeus is the warm dead. Those are samples that are in museums at room temperature. So this sort of gives you a way to sort of think about the whole landscape. And the 2 here on the, what is this? This is left, right? Is life on ice. That's sort of the tagline. Now, if you think about life on Earth, here's a tree of eukaryotes, and what does it mean to preserve the global genome? Essentially, it may mean, and could mean technically, has often been taken to mean technically, the sum of the branch lengths of the tree of life. In which case, is this like this guy right here, Opusozoa, is a sister group to all of Metazoa. So if you think about preserving it, what you want to do is spread your effort across the tree of life, getting as much as you can. It's an open and interesting question whether genetic diversity is concentrated at the tips, population genetics, or whether it's sort of bulges here in the middle where major apomorphies and adaptations evolve, or down in the bottom. We do know with ortholog DB that there are a whole bunch of housekeeping genes that are common to practically all kinds of life. So why is this feasible? Let's take families, which is a relative term for a clade of a certain size. And this is from Linnaeus forward for a couple of large groups of organisms. And this means biological discovery, not knowledge. So this means, when is the first time that someone went out in the woods, picked up this animal and said, damn, that's a new family? Technically what I mean by it's the earliest date of a species that is now classified in this family. And the point about it is that it's largely asymptotic. People discover new families, in that sense of the word, somewhere between 5 and maybe 15 times a year. And there are about 9,911 families on Earth. So we actually know, at that level of clades, what we're doing. We know what's on Earth. There is no sort of deficit of discovery as there is at the level of species, where we don't even know to an order of magnitude how many there are. Families, as far as we can tell based on this historical record, are essentially known. So if we then look across all of the big data sources that you have for all of life, so this is GBIF, Biological Heritage Library, GenBank, Open Tree of Life, BOLD, Encyclopedia of Life, GGBN, which is an organization I'll describe in a minute, we have about 1.1 billion data points in which the strings for the family, the text strings for the family, show up. That's a very high density of data. And if you look at the number of families present, you can see, so there's 9,911 families. That's sort of a reference taxon. There are many other taxonomies, but that's the one I'm going to use. Only 196 families are absent from all 7 DBs, which is extraordinary. It means that we know what we're doing. With respect to BOLD, there's about 4,200 families of life that do not have barcodes. If we want to enable approximate identification to the level of families, why not prioritize those 4,200 families? And you can sort of go to the world and say, we're done at this level. Any kid anywhere on Earth can, barcode this and get an approximate family idea. The interesting question for all of us is, will that work? If I have one barcode in every family, what's the success rate of pinning that unknown to that family? So we have a lot of data. Now, when you look then at where those, say, phyla, there are about 101 phyla. This is always be the same, 101 phyla in the catalog of life. Then we want to know, for example, GenBank and barcode over here. You can see that there's 37 phyla that have been barcoded. That means there's still a lot out in the wild. Do we have tissues and do we know where there are? There are about 68 there. Global Catalog of Molecular Organisms is sort of a sister organization to us. They have 57. There's 16 that are still out there. We know they're there, but nobody's gone out and gotten them. Here's a list of those. And most of them are marine invertebrates or parasites, but there are some things like onychophorans. They've already published genomes of onychophorans, but as you know, people who do genome projects never deposit the tissue or declare a voucher. They just publish the genome, and it's sort of, vaporware. Another interesting one is hornworts, one of the early branching lineages of plants, also not there. So to me, as a field biologist, this is exciting because it gives you 16 things that you have friends somewhere on Earth, and they know how to do this. We just haven't activated that kind of priority setting. If you look at the level of families, the number here varies a little bit. This is straight from the catalog of life, so it's a little bit less. There's 5,000 that are still out there that we know they're there, but nobody's done anything about it. There's about 3,500 in GGBN, 3,200 over here. So what you see is that as you go down the scale, the white space gets bigger. talking about genera, I don't even do this at the level of species, partly because we don't know how many species there are, but there are about 160,000 described genera of life on Earth, of which, interestingly, about 105,000 are insects. So 2/3 of the valid genera on Earth are insects. That sort of justifies why Guelph is in the insect business, for sure. There's about 25,000 here. I would say if we got half of those, most of the identification questions that people are interested in asking would be answerable. There's a long tail of really rare and actually even almost imaginary genera that have been described in the 1800s that probably are not valid. But if you really just want to fire up the ability to do biosurveillance, this is the interesting battleground, is genera. Families we can do, I think. So GGBN, the Global Genome Biodiversity Network, prior to its existence, what we had was GBIF that did specimens and vouchers. That was good, Darwin Core Archive. You had NCBI that did sequences, but there was never a connection. Nobody did a proper data model for tissues, DNAs, RNAs, physical genetic resources. It was the missing link. So you could have a sequence, but you didn't know where the tissue was, you didn't know what quality it was, you didn't know who had it, you didn't know where it came from, you didn't know how it was vouchered. So GGBN is a relatively new organization that sort of is the missing link that connects sequences to the warm dead, so to speak. Now, it's about 3 or 4 or 5 years old. We're up to 83 members in 30 countries, 2 million samples, about 3,900 families, 20,000 genera, 45,000 species. Here are some of the 83 logos. And I would like to invite any of you, there are 5 institutions in the audience who are already members of GGBN. That's Edinburgh, von Humboldt, God, I shouldn't have tried to remember them all while I'm giving a talk. Oh, us, and Guelph. So this is a way to mobilize your collections and make them machine reasonable across the whole globe. And with 27 countries here, 17 countries here have GGBN members in their countries. So it's also designed to thwart the impact of Nagoya of moving tissues across international boundaries. If you have an institution in your country and you're not in the collection business, give the tissue or the DNA to them. It'll work. Now, the core thing that we did was to develop an extension to Darwin Core, which is for tissues, DNAs, genetic resources, sequences, amplicons, extracts, and it has sort of 5 major categories of fields. There are 156 fields in it. So this is battle ready for 21st century biodiversity genomics. It handles the link between genetic sequencing and tissues and vouchers and everything else. Importantly, it also is battle ready for Nagoya. So it's taken into account what we can anticipate about the necessary sequences to prove that you have the permission. 2 minutes. Yeah. Should go. So this is sort of our progress to date from kingdoms, phyla, classes, orders, families, and genus. Blue is what we've got. Gray is what's out there. Now, just one comment about the sequencing. We heard just the previous talk. We'll hear some more about it. There's about 3,300 eukaryotic genomic samples on Earth that have been done. And this is sort of you can either do a genome level of 4. The 4 is the complete, that's the first digit down here. Chromosomes, scaffolds, and contigs. There are other kinds of nerdy ways to classify genomes. But you can see that this is sort of where we want to be. This is sort of where we are now. This community is going nuts because access to high quality tissue is absolutely rate limiting for them. So Paul mentioned $1 billion for iBOL 2, the Earth Biogenome Project. I'm part of the working group. They figure it's $4.7 billion. So I'll see you $1 billion and raise you 3.7. And half of that is scheduled for sample collection. That's an opportunity for our community. The other thing, maybe I don't have time to do this, but nobody really knows how to measure high quality DNA. This is sort of in the 150,000 base pair fragment length. You even get into things like pipette shearing, where when you push the DNA out to the end of the pipette, it tears into fragments. So you have to hand trim your pipettes, and the people who are doing, cutting edge biodiversity genomics do this kind of stuff. So, summary, big data, we've got a lot. Biodiversity discovery at a certain level. We are equipped to do what we want to do. Preserving the genome might be the sum of the branch lengths. And then this gap analysis, knowing what's out there, sort of the known unknowns, can inform the strategy of how we ramp up our mega projects to provide early payoffs so that we maintain our credibility. Thanks.